Hello students, welcome to medicoRAS. Today we are going to discuss some of the questions that I got from the students. So I hope everyone will get benefited by seeing these questions. So let's start with the first question. Question number 1. You can see here they are mentioned that a tobacco plant is a heterozygous, and for albinism the character is recessive. And after self-pollination, that heterozygous plant is actually giving 1200 seeds. Subsequently, it is germinated. Then they are asking that how many seedlings will have in the parental genotype. So what they are asking that we have to go for self-pollination first, and the plant is heterozygous. So if I consider albinism as a recessive character, that means I can consider it as something like this. So now if I consider that both the plants are heterozygous, because I have to go for self-pollination. So in this concept, the genotype of the parent will be something like this. Now you can see here that from this I can get this capital A and small a as a genotype. Similarly, here also the same concept, capital A and small a as the genotype. And if we do, this is our gamete, right? This is our gametes. And this is what parents. Now if we do cross among all those gametes, then what is the result? Result will be something like AA, capital AA. Then again A combined with, if it is small a, then it becomes capital A and small a. If A combines with again capital A, then it will be again the heterozygous entity. And if I go for this small a, then I'll get the homozygous, that is albinism. Now you can see here two progeny, they are showing what parental behavior. Now they are asking how many parental genotype we can get from this type of cross. Here you can see that we can get 4 different numbers of progenies. So out of 4 number of progeny, 2 number is showing what parental behavior, parental genotype, and total how many seeds we are getting, like 1200 seeds we got. So if it is something like this, then we can calculate it easily and we can get the result. It is nothing but the 600. So I hope you understood this one. This is very simple concept that we need to find out the parental genotype from the progeny, right? So next, come to the question number 2. So in this again cross pollination occurs and this much plant we are going to produce, or it is already produced in the next generation, fine, from the 2P plant of course, and out of which 94 plants were tall, 89 plants were dwarf, right? So we can consider, if we calculate 94 plus 89, we can get 183. That means they have given the result of this one from this two combination, right? First and foremost, you need to know whatever the plants we need to get, or whatever plants we got already, that is actually the result of the concept or not. Yes. So from this I can easily understand that whatever the result they have given, from that only they have mentioned that this much plant we got. Now you just see 94 and 89. There is hardly a difference of 5. Right? So we can consider it as 50% and 50%. That means 50% plant were tall and 50% plant were dwarf. So in that case, just you look all the options. We need to find out the parental genotype. So you look all the options, in which option we can get this 50-50 concept. Right? Now, can you see here the all parents, the two parents are actually what? Dominant. There is no concept of recessive. So we never can get the dwarf concept, right? So we can't take this one. Again, if you see this, then here only one parent is capital T and small t, but the other parent is capital T, capital D. That means small t is absent in the other parent. So that's what we can't take this one, because for dwarf we need small t 2 times. Fine. Again, you see here we have a little hope that we can get the answer, but condition is that if we go and do the cross, then only we can get this type of progeny. That means we are not going to get two progeny, tall and dwarf. We are getting only one progeny, and that is tall. So this option is not applicable. But we can see in the last option that there is a concept that in both the parents, at least I have small t and small t one time, at least, right? In the other parent, no doubt there is two small t, but in the first parent, we can see on at least one small t is there. So I can go for the cross here, and I can find the gamete. Let me write it here so that it can be easily visible for you, everyone. So see, these are the two gametes, and from here I can get this type of gamete, right? Small t gamete. And if we cross these two, then you can see here that only two types of progeny I am getting, out of which one is tall and other one is dwarf. That means it is 50-50 concept, correct? So if it is 50-50 concept and I am getting tall and dwarf, then obviously the answer must be this one. Yes, so here option B is correct. I hope everyone understood. So let's go for the next questions. Come to question number 3. In our DNA, number of hydrogen bond is 700 with 10% of adenine, and we need to find out the length. So for that, we need some concept from the DNA helical structure. Yes, so we know in the DNA helix, from one pitch to the other pitch, that means in complete one turn, we have total of 10 base pair, and that is the length of 34 angstrom, correct? Again, we also know that if, according to Chargaff rule, adenine is always equal to thymine and guanine is equal to cytosine. So if adenine is 10%, then obviously thymine also have the 10% concept, correct? So 10 plus 10, total how many percentage we got? 20%. So out of 100%, if 20% is only for adenine and thymine, how many left? Now 80% left. So that 80%, if I equally divide, then 40% will be for the guanine and 40% will be for the cytosine. So this is the basic concept that I obtained from the helical DNA helical structure. Now let me consider that in that 10 base pair means what? 10 base pair means 20 nucleotide, right? Because one base pair has 2 nucleotide, so 10 base pair has 20 nucleotide. So out of that 20 nucleotide, fine, if we have 10% as adenine, that means how many total numbers of adenine nucleotide we have? 2 numbers of adenine I have. Understood my point? And like that, I have thymine number is also 2, correct? That means what I can say that if it is 20 nucleotide and 10% I have given for the adenine, 10% I have given for the thymine, then obviously remaining out of that 20%, yes, remaining 40% will be for cytosine, and that is nothing but what? 8 numbers. And you can calculate similarly for guanine, it is also 8 number. Just to calculate everything, you will get 20 numbers of nucleotide. That means our calculation is till now correct. Now you see here we have adenine 2 number, right? Adenine 2 number, thymine 2 number we can see here, correct? So we know adenine and thymine, these two are actually bind with double hydrogen bond, yes, and guanine and cytosine, they bind with triple hydrogen bond. So if I have 2 numbers of adenine, that means how many hydrogen bonds I have? For 1 I have 2 hydrogen bond, correct? So for 2, how many total hydrogen bonds I have? 4 hydrogen bonds, correct? So similarly, if I go like this, then I can find out all the hydrogen bonds which are present here, right? That means total how many hydrogen bonds? See, these things I can calculate for the let me say this is for adenine, then I can calculate the same concept for the thymine as well. So it is 4 hydrogen bond for thymine as well, right? Because thymine also have 2 numbers, right? Again, if I go for cytosine, then 8 into 3 hydrogen bond, because we know that thymine we know cytosine and guanine, they are bind with 3 hydrogen bond. So I have to write 3 hydrogen bond here. for one cytosine. So if I have 8 cytosine, how many hydrogen bonds I'll have? For 24 hydrogen bonds I have, right? Similarly for guanine also I have the similar concept like this, yes, and therefore I will get a 24 hydrogen bond. But you can see here, if I have written 4 hydrogen bond, that hydrogen bond is applicable for the thymine itself, because adenine and thymine are going to bind, right? So that means 4 hydrogen bonds are actually present in between these two. Again 24 hydrogen bonds will be present in between these two. So no need to calculate two times. Just I'm showing that we are having the same concept for these two complementary sequences. That means we can calculate only one strand of nucleotide, right? That is enough, because the other strand will have the same concept. So if I calculate only the one strand, that means 4 hydrogen bond and 24 hydrogen bond. Total how many hydrogen bond I have? 28 hydrogen bond I have. And we are considering what? 10 base pair. So for one strand, how many hydrogen bond I am getting? 28 hydrogen bond. And what is the length of that hydrogen bond is what is the length of that DNA is 34 angstrom. Then you just find out with the unitary method that how many what is the length if the hydrogen bond number will be 700, right? So simple calculation, it is 34 into 700 by 28. Answer as 850. So what is the answer here? Option D. So 850 angstrom is the answer. I hope you understood this part. Yes, if you don't understand any of the question number or any of the point, please mention in the comment section below. I'll get back to that question again. Okay. Now, can you see here we have another question, and that question is question number, actually it should be 4, right? Now, see, in a resistant to a fungus, pea plant is conferred by H gene, H, small h they are representing, right? So, resistant gene is actually what? That means recessive. Please remember this concept. Resistant gene, what we have? Small h concept, right? Recessive. Already they have mentioned. It to its allele, and that is H, capital H is for the susceptibility. Okay, so if it is capital H, that means susceptible. Yes, susceptible gene, for that I may have two concepts. This one I may have, or this one I may have. Then let's see the other portion of the question here. If a resistant female, yes, if a resistant female, that means the female must be this one, right? So let me write here, the female is crossed with a homozygous susceptible male. So if it is homozygous, I couldn't consider this one, right? I have to consider this one, so it must be the male. Now, what would be the order of genotype in the pistillate female? Pistillate flower or pistillate parent means what? Female parent they are asking. And what will be the genotype for the staminate or the male parent, as well as male gamete and the egg? You can see here already we got the answer that female genotype must be small h, and male genotype must be capital H, capital H. So from here, if I say egg will have what type of gamete? Only small h. And if it is sperm, or if it is like pollen grain, if it is a plant, then obviously I have to write what? Pollen grain. So in that concept only I am getting this capital H as a gamete, right? Now I have to write male gamete first and egg second, right? Again, pistillate flower I have to write first and then staminate flower. Then see, this is the answer, right? Here the pistillate flower is capital H, capital H, small h, sorry, and male is capital H, capital H. Then male gamete is capital H, and then egg has small h. Already we got all those answers. So what is the correct answer here? C is the correct answer, right? Very simple concept. Just you need to read the lines, and you need to find out, you need to write everything, and then you will get the answer, right? Next, let's go for the next question here. So this is the last question for the session, that is question number 5. So here they have mentioned that consider a monohybrid cross between a pure tall, yes, and a dwarf plant. Okay, pure tall means this one and dwarf plant means this one. Fine. In F1 generation, of this cross, 4,000 seeds already it is produced. How many seedlings will be dwarf? See, we have this concept. So already we did this type of question, right? So here I can see that if this is the parent one, then from F1 generation, I can get only this type of progeny, which is purely tall. Then if I go for the selfing, this is selfing, right? So if I go for the selfing, then I will get in F2 generation, capital T, again 2 capital T small t, and I will get this one, right? Just now we have did this type of question, correct? So out of which they are asking that we have how many seedlings will show the dwarf concept. So we can see here this is tall and this one is also tall, which is heterozygous tall of course, and this is what dwarf. That means out of 4 numbers of progeny, how many number of progeny is showing a dwarf concept? Only 1. That means I can write 1 by 4 is the dwarf. And total how many seedlings we got? That is 4,000 seedlings. So if we calculate like this, that means we will get 1,000 number of seedling as a dwarf seedling, right? So what is the answer here? This B is the answer. So like that, we are going to take all the questions from different chapters, that is from the students' side. So I hope everyone understood all those points. If you have any doubt, query, you just write in comment section below, and I'll definitely take those type of questions, or I will try to clear your doubts. Thank you so much.